All right, welcome back. This is organelles part 2. So the next set of organelles we're going to talk about are only found in eukaryotic cells. So we're talking plant and animal. We will talk about if any are found in only one or the other. Okay, so first organelle we are going to talk about is the endoplasmic reticulum, or ER. So this is a diagram of the ER, and you're going to notice we're going to talk about both a smooth and a rough ER. So first let's talk about what these are. So the endoplasmic reticulum is a network of membranes. And every time you hear the word membrane now, you will think phospholipid bilayer. Okay, so this is a continuous. It's hard to spell sometimes in front of people, even though I'm not in front of anyone. Continuous network of membranes, and what I want you to see is it is continuous with the nuclear envelope. See that right here? So it goes from nuclear envelope, and that develops right in to the ER. So they're all interconnected. Usually when I draw it, I would have my nucleus here, and then I just kind of draw these like, that's how I diagram the ER. So you can kind of see that. We have a rough and a smooth. So the rough ER is rough because it has all these little dots on it, which are ribosomes. So it has ribosomes attached. So hopefully you go, ding, 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 ribosomes, site of protein synthesis. Rough ER must also be a site of protein synthesis. So the rough ER is in fact a site of protein synthesis. And we will actually talk later this semester that proteins can be made on free-floating ribosomes and on the rough ER in eukaryotic cells. The rough ER is also not only a site of protein synthesis, but also, let me go back to red so we keep this all together, and protein modification. And we're not really going to talk about this much this semester, but it's really important when you take cell biology. We'll talk a lot about protein synthesis. I think you probably also cover it in genetics. Okay, so let's contrast that rough ER to the smooth ER. Okay, so the smooth ER is smooth because you can see there are no attached ribosomes. The smooth ER is very important for, I'm going to put function. Sorry, I write it aslant because my tablet's like not flat, so I'm having a hard time figuring out how to write, but I'm sure by the end of the semester I'll be awesome. The function is to make lipids, and carbohydrates, and we'll talk about all these macromolecules in another week or two. It makes some hormones like estrogen and testosterone. It also has a role, so it's a producer I'm going to put up here. It also has a role in what we call detoxification. So your liver, the cells in your liver are full of smooth ER, where a lot of drugs, alcohol, other foreign molecules come through and get detoxified by the smooth ER. And the detoxification is so that we can get those chemicals out of our body. In the muscle cells, and in other cells, but especially in the muscle, there's a storage of Ca2+, which you may or may not know is calcium. So calcium ions are also stored in the smooth ER. What's really super cool is that these two, being continuous network of membranes, they can kind of change. So smooth ER can become rough if the cell needs to make a whole lot of proteins, then more ribosomes will start attaching and it will become rough ER. Or, like I said, in the liver, if you really need to work on detoxification, you'll lose ribosomes and the rough ER will become smooth ER. Pretty cool. Again, eukaryotic cells, both plant and animal, and these are just some TEM micrographs of the smooth and the rough ER. So this one hopefully you recognize as a rough because it has all those dots on it. This one is smooth. And it's interesting, they kind of change shape. So you can see that the rough, this is why I draw it like this, you know, it's just this back and forth network. And then the smooth seems to be more tubular. And that just is based on how they're working, their function. Okay, so this is near the nucleus, right? Continuous with the nuclear membrane. The next organelle in all eukaryotes, plants and animals, is the Golgi apparatus. Okay, this is the one organelle that you must capitalize because it's named after the scientist that discovered it, Golgi. This is different. This is a series of individual stacks of sacks. Series, let me just write it. Series of individual sacs. Or membrane sacs, I should say. Okay, so every line you see here, that's your membrane. And what I want you to see that's different is that these are not connected. Okay, they're individual, so it's not a network like like the ER. And you can kind of see that. Here's a TEM, and you can see, I should change color so you could actually see what I'm drawing. You can see all these individual sacs. So some people say it looks like a stack of pita bread or a stack of pancakes. The function of the Golgi is to sort, package, tag, and distribute. Okay, so let's write that. Sort, package, tag, and distribute proteins and lipids. Hopefully you can read my writing. The Golgi, some people describe it as the UPS of the cell. So it gets, you can see incoming, it gets stuff usually from the ER, and it will sort it, it will maybe tag it with sugars or other modifications so that that molecule knows where to go in the cell. It will package it into these vesicles, which is our next term, and distribute it to the right parts of the cell. Again, yep, all eukaryotic cells, plants and animals. So let's talk about these little vesicle things. So we have two terms here, vesicle and vacuole. So both are membrane sacs. Okay. So we got lots of membrane sacs. So remember, eukaryotic cells are full of what we call membrane-bound organelles. So we have membranes so that we can have distinct functions in all of these different organelles parts of the cell. Remember, that also allows the cell to be a little bit bigger, or not a little bit, a lot bigger than a prokaryotic cell, because it has all these specialized organelles in it. It helps move things around better. Vesicles in general are for transport. Okay, so moving things from one place in the cell to another. So you can see down here from our rough ER, we have a transport vesicle that's bringing, say, some proteins to the Golgi. And then from the Golgi, we have what we call a secretory vesicle that is going to take these proteins to the plasma membrane to secrete them. So sometimes vesicles will have these special modifier names, transport, secretory, things like that. Again, you'll really get into this in cell biology. Vacuoles in general are more for storage. So the one vacuole that I want you to know about is the central vacuole, and this is only in plant cells. Now, there are all kinds of vacuoles in animal cells, plant cells, protists, I'm guessing fungi, I don't really know. But we're just going to focus on subsets here. So the central vacuole in plant cells, is for the storage of water. So it's huge. It takes up a huge, it's hard to see here, huge space in the plant cell. And what's really cool, so if you've ever, and I wish I had videoed this the other day. So I have this big planter full of catnip for my cat, and he goes out there every morning and munches on it and then like passes out. And I went out the other morning, and one of the containers, it was all wilted over. We had forgotten to water it the day before. And I was like, oh man, I just lost all this catnip. I thought, well, I'm just going to try it. So I gave it, sorry, I've been not moving around in my office now. I gave it a whole bunch of water, and these wilty, like I really thought they were dead plants, sucked up that water, came back to life, looked beautiful, and that's all the central vacuole. So that central vacuole has to fill up with water and make the plant cell like super full so that the plants can stand up. So, like I said, I thought these guys, these plants were gone. I wish I had videotaped. Maybe I won't water them tomorrow and video them, post it for you. Okay, so we've got vesicles, vacuoles. Both are membrane sacs, slightly different functions. One's for transport, one's for storage in general. Vesicles in all eukaryotic cells. Peroxisomes and lysosomes. Wow, guess what these are? These are small sacs. These are membrane sacs too. Okay. But these, inside the membrane sacs, they have enzymes. And enzymes are a type of protein we'll talk about in another week or so. And these enzymes make chemical reactions happen. So they both have membrane sacs with enzymes, right over here, to break down different molecules in the cell. And by break down, think like digest. so that we can recycle, reuse parts of the cell or molecules in the cell. Okay, I would never ask you to look at a picture and decide which is a lysosome, which is a peroxisome, which is a vesicle, because they're all membrane-bound sacs. They look very similar under the microscope. These two have slightly different functions. So lysosomes, they use enzymes called acid hydrolases, and they can break down things even big like old organelles. So if you have we haven't talked about mitochondria yet, but you probably heard that term. So if you have an old mitochondria that's not working well, a lysosome will surround it and then break down that old mitochondria, and we can reuse the parts. So it can break down old organelles or material from outside the cell. So if we go back one slide, you can see that this is a picture of what we call a macrophage, and that's part of your immune system. And what macrophages do is they go around your body and they're looking for foreign objects like bacteria. And so what this macrophage did was it took its membrane and made a circle of membrane, made a vesicle around that bacteria. And then here comes the lysosome, which is another membrane sac, but now it has enzymes in it. And these two mush together. Remember I told you all these membranes, just like bread dough, Play-Doh, can mush together. And now you're digesting this bacteria into all the little pieces. So you're destroying it, saving your body, and then you can reuse parts of it. So that is, I kind of made these opposite. That's a lysosome. A peroxisome works in detoxification, different methods than the smooth ER. It also breaks down fatty acids, and amino acids, which are parts of proteins. So the peroxisome here is just a little bit more specialized, but they're both functioning to break down molecules, and they're both membrane sacs with enzymes. Okay, this is where there's a thing to note. So peroxisomes are in both animal and plant. Okay. Lysosomes you find in animal cells. Lysosome function in plant cells are in a vacuole. So, like I said, vacuoles are for storage pretty much, but the more you learn about biology, the more you learn that any kind of rule you try to make, it gets broken by something. So, plant cells, let me just write this down here. Cells use a vacuole, not the central vacuole, just another vacuole for the lysosome activity. So they can still break things down. They just do it a little bit differently. Okay, I think I got all of that. So the endomembrane system is basically the organelles we were just talking about. So it consists of the nuclear envelope, the ER, the Golgi, these vesicles, the lysosome, and the plasma membrane. So there's what, 1, 2, 3, 4, 5, 6. 6 parts. And this is the figure I just showed you earlier. This system works to make, modify, sort, distribute all of our proteins and lipids. So it works together to create all these molecules that we need in our cells. So that is the endomembrane system. Endo means inside, membrane system. And one of the reasons this internal membrane system works is, like I said, you can have vesicles. Let me do a different color. You can have vesicles bud off from, say, the smooth ER and go and smush into the Golgi and then bud off and go where it needs to go. And this is a YouTube link from your textbook. It's pretty good. Just walks you through the endomembrane system. Okay, we have one more kind of classification of organelles that we're going to talk about, and that is the cytoskeleton. This again is in eukaryotic cells. And these microfilaments, intermediate filaments, microtubules, these are all protein fibers. And we'll talk about proteins in the macromolecules of the cell, but hopefully you've thought about protein, right? You need protein in your diet. Proteins are one of the most diverse types of macromolecules as far as functions in the cell. So I mentioned enzymes are proteins. Now you know that the cytoskeleton is also made of these long protein fibers. You've got three types, and we're going to talk about each type in a second individually. But in general, what the cytoskeleton does is it maintains the cell shape. It helps for cell movement. And it just kind of keeps everything organized. So it keeps different organelles where they should be. We'll talk about in just a minute, microtubules act kind of like train tracks, so things can move in orderly fashion. Prokaryotic cells do have a type kind of cytoskeleton, but it's nothing like this. So when you're filling out your chart, we say that prokaryotes do not have a cytoskeleton, but plant and animal cells do. Okay, and this is an image showing you the different types of cytoskeleton. So here in A, you have what we call microtubules, and like I said, I'll talk about the individual functions in just a minute. These actin ones are called microfilaments, and this one in yellow is called an intermediate filament. And what I want you to see is that the microfilaments are the thinnest, the microtubules are the thickest, and gosh, guess what? The intermediate are right in the middle there. Okay, so let's talk about the functions. So, microtubules. These, if you were going to describe them, they are hollow tubes. Right? Remember, made of protein. Some of their functions in the cell, they're like the train tracks. So for moving things around in the cell, there are these really cool things called motor proteins that will like hold organelles and vesicles and walk down these microtubules back and forth to take things in an orderly fashion from, say, the ER to the Golgi. So train tracks in the cell, and you can see these are all stained green. It's really pretty. for movement of vesicles and organelles. I should put train tracks. Microtubules also make up what we call the spindle fibers that are important in pulling chromosomes to separate cells in cell division. Whoa. Okay, so spindle fibers for separating chromosomes during cell division. Microtubules also make up something we're going to talk about in just a second. They also make up a thing called a centriole. They make up flagella, which we'll talk about in the last video. And cilia are made of these hollow tube proteins, microtubules. The next one we're going to talk about is microfilaments, the skinniest one, and these are shown in red. And sometimes they're also called actin filaments. And they're really important for cell movement. So some cells in your body, like white blood cells, actually can crawl along, finding foreign objects, part of your immune system. So cell movement, excuse me, oh, sorry, actin filaments or microfilaments are very important for. They are super important in your muscle cells. So this is how your muscles contract, actin and myosin. They are also important in dividing the cell, so in dividing the cytoplasm during mitosis and meiosis. They really help. You can see they're all over the place with cell shape. And microfilaments are important for cytoplasmic streaming. So if you don't know what cytoplasmic streaming is, it's really super cool. Go click on this YouTube video. You can see it really neat in plant cells under the microscope, so live cells. And you see the chloroplasts kind of moving around in this really organized fashion in order to get the best light for photosynthesis, which we'll talk about in the last lecture. So check that out. It's really cool. Okay, that's our microfilaments. And then, and so a description of these, did we do, yeah, we said hollow tubes. This is 2 strands of actin intertwined. That's how you would describe it and recognize it. So if you see these just two strands, that's a microfilament. If you see something that looks like a hollow tube, that's a microtubule. And then if you see something that looks like a whole bunch of strands together, so this is multiple strands twisted together, that is an intermediate filament. Intermediate filaments are important for cell shape. They're important for cell structure. They give the cell strength to reduce, to not like fall apart, and to help in stress, like if your cells are getting smushed or stretched. So cell structure, cell strength, Really super important for the structure of the nucleus. And there are lots of different types of intermediate filaments, including like what makes up your nails, things like keratin. But what's really, I think, fascinating is that there's different types of intermediate filaments in different cell types, so in different tissue types in your body. And one of the ways they can figure out if you have cancer that has metastasized, so that means cancer that has moved from where it started, they actually analyze the intermediate filaments, and they can tell where that cancer came from. Did it come from your liver? Did it come from the breast tissue? Did it come from, I don't know, wherever cancer comes from? They can tell that by the specialized intermediate filaments. I thought that was cool. Okay, last slide before we stop this lecture, centrioles and the centrosome. So I mentioned that centrioles are made up of microtubules, and these are essential for the division of chromosomes. So a centriole is actually made up of a whole bunch of microtubules. So each of these rods is a microtubule that comes together to make that centriole. These are only found in animal cells. And they're always found in pairs. And they can actually replicate and divide. And what their function is you can hardly see them. Let's see if I can. I never can zoom in when I want to. I only zoom in when I don't need to. Okay, never mind. The centrioles are right here, and there's this area called the centrosome. So centrosome is an area where the centrioles function. And what is made from these centrioles is these things called spindle fibers. Uh-oh. Oh. These spindle fibers are also microtubules, and what it's trying to show you here is that they're connecting to these replicated chromosomes, and as the cell divides, the spindle fibers pull the chromosomes towards the different centrosomes, and then the cell will split down the middle. So we'll discuss this more when we talk about mitosis and meiosis. Here's an electron micrograph. You can see the centrioles right there. Okay, I'm going to pause, and we will come back and finish up with cell wall, chloroplasts, mitochondria, endosymbiosis, flagella and cilia, and fimbriae and pili. So we're kind of now going to the outside of the cell. All right, I will see you at the next lecture.